Welcome back, dear students. Now we're talking about epistasis, and we mentioned that when different genes which are not allelic to each other, and if they affect the same character in such a way that the expression of one will mask, inhibit, or even suppress and sometimes modify the expression of the other, it is referred to as epistasis. And epistasis could be of two types, dominant epistasis as well as recessive epistasis. If the gene which is masking or modifying the expression of the another non-allelic dominant gene is a dominant gene, then it is referred to as a dominant epistasis. And if the gene which influences the expression of a non-allelic gene, another non-allelic gene is recessive. Okay. In such a case, it is referred to as recessive epistasis. And we also found that the kind of epistatic interactions can be found to be different types. Like sometimes it might be dominant epistasis, recessive epistasis, or sometimes you can also find duplicate recessive genes, duplicate dominant genes involving, as well as duplicate genes with cumulated effect, as well as dominant recessive interactions. In this session, right now we're going to talk about the dominant epistasis. If we look onto dominant epistasis, the epistatic gene, which is being considered over here, is a dominant gene. And this has been found in various plants as well as animals. But here, we mainly consider an example of summer squash or the cucurbita pepper. This is a fruit which can occur in different colors like white, yellow as well as green. And the white is dominant over the colored squash. And yellow is dominant over green squash. So if you're going to consider the level of dominance, W is more dominant and then comes yellow, and below that comes the green squash. And pure breeding of the white fruited variety, okay, is crossed, okay, with the double recessive green variety, and the F1 hybrids are all white when we cross, okay. So you are taking, when a white hybrid is taken and it is crossed with a green, a pure breed of white is crossed with a pure breed of green variety, and you get a F1 hybrid which is considered to be all is white. And when the hybrids are being selfed, what happens? White, yellow as well as green fruited plants arise in the ratio of 12:3:1. Now just have a look onto that. Okay, these are the different varieties of the cucurbita. And so what happens is, as I told you earlier, we are taking a dominant, as we mentioned earlier, the white one is dominant. And when you take a proper pure breed of that, it will contain WWYY. And if you take a pure breed of the green variety, it is small letter WW and small letter YY. And the thing that which we should know is the dominant gene W here, okay, is considered to be as the epistatic gene. What is the epistatic gene? Epistatic gene is a gene which will influence the expression of the other. Now, is it an allele? What is the difference between an epistatic gene and an allele which influences the other? In this case, what happens? The expression of another gene, that is a Y gene, is influenced by a non-allelic gene, which is the W. So W and Y are not alleles. They are two different genes over here in this situation. And the allele of W, capital letter W, is small letter w. In the case of Mendelian ratios, the concept of dominant and recessive was being given for two different alleles. That is, if you have capital letter W is dominant and small letter w is recessive. But here, in the case of epistasis, we can see that the expression of one gene, Y, okay, that is being controlled, or it is being masked or inhibited or sometimes modified by the help of an epistatic gene or a epistatic non-allelic gene W. So that is a case of epistasis. Always non-allelic interactions are happening in epistasis. And in this example which we have here, we are having a purebred white fruit with a purebred green fruit, and the green fruit is being found to be recessive in nature. And as a result, you get a hybrid which is being considered to be capital letter W, small letter w, capital letter Y, and small letter y. And of course, due to the presence of capital letter W is over here, as I told you, capital letter W is a white one and it is dominant over the Y gene, and as a result, what happens? A white fruit is being obtained. And when this hybrid is being selfed again to get the F2 generation, you get different types of fruits. You will get white fruits, you will get the yellow fruits, as well as you get the green fruits. And of course, this is quite clear to you, the Punnett square and the different gametes which are possible from it. If you have WWYY, we have W, capital letter W and capital letter Y, capital letter W and small letter y, and small letter w and capital letter Y, and small letter w and small letter y. The same gametes will be considered here because we are in the F2 generation, we are crossing F1 against F1. So as a result, the different hybrids which are being formed are being shown over here, or the different offsprings which are resulting after the in the F2 generation are being shown here. And you can see here, in all the white varieties, we can see that there is a capital letter W. A capital letter W is being present over here. Okay, and if you're going to take the green color one, okay, the green color one, you have small letter WW and small letter YY. Okay, so it means that over here, this is a recessive trait over here, and only when the capital letter W was coming, okay, it is inhibiting the color yellow as well as green over here. Even in the case of, you can see, in the case of yellow also, the expression of this yellow was being inhibited here when you go to consider the white ones. Even if capital letter Y was being present in some of these, what happens? Because of the presence of a capital letter W, what happens? The effect of capital letter Y was being inhibited. But if you go to look at the yellow fruits, you can see that they are all conditions in which the genotype lacked a capital letter W. That means the W gene was being found to be recessive over there. Only in such conditions the capital letter Y was being able to express itself. So this is a case of dominant epistasis, where a dominant non-allelic gene will influence the expression of another dominant non-allelic gene. In this case, we have W, which is a dominant epistatic gene, and capital letter W, of course, and capital letter Y is a dominant hypostatic gene, as we had mentioned earlier. You know what is the difference between epistatic gene and hypostatic gene? The one which influences the expression is referred to as the epistatic gene, and the one which inhibits or which gets inhibited is referred to as a hypostatic gene. So Y, capital letter Y here, is considered to be as a hypostatic gene. Now, if you go to take small letter w and small letter y here, what happens? The expression of Y is not inhibited over here, but at the same time, you're getting a different version. You get a green color type of fruit. And the phenotypic ratio in this example, as we have noticed, we have 12 white, 3 yellow, and 1 green. So the phenotypic ratio of 12:3:1 is observed in dominant epistasis in this particular example. Of course, you can have different ratios with different organisms, but commonly speaking, dominant epistasis is a condition where a dominant allele of one gene will mask the expression of the alleles of another gene which is non-allelic. We can also see similar examples of dominant epistasis in white leghorns, which are white, as well as Plymouth Rock variety. When we cross them, you can see similar patterns of dominant epistasis also. I have not included that example in this session. I guess this is clear to you. Thank you for now.